So, what if the very same technology that could create our most effective vaccines could also, in theory, be used to turn our own immune systems against us? Well, that's the paradox at the heart of epitope manipulation, a really powerful capability in modern biotechnology that we're going to explore today. Yeah, this is really a story of two sides of the exact same coin. On one hand, you have the power to engineer incredibly precise, effective vaccines. But on the other, you have the theoretical ability to flip that science completely on its head, turning our body's own defenses into a weapon against itself. So, how can one single toolkit lead to such wildly different outcomes? Let's break it down. Okay, here's our roadmap. We'll start with the dual-use nature of this tech. Then we'll dive into part one, how it's used to engineer immunity. After that, we'll look at the darker side in part two, engineering disease. And finally, we'll see how these two paths all come together. All right, so let's start with the incredibly promising side of this technology. We're talking about how we can use it to engineer immunity and build way better, smarter vaccines. So here's the central problem in a lot of modern vaccine design. When a pathogen gets into your body, your immune system can actually get pretty distracted. It often wastes its energy attacking these decoy epitopes, parts of the pathogen that don't really do anything to stop the infection. It's kind of like your immune system is shooting at the armor plating of a tank instead of its engine. It's not going to do much to neutralize the threat. So how do scientists solve this? Well, the answer is a brilliant little molecular tool called glycosylation. By engineering a very specific sequence of amino acids into a protein, they can basically tell a cell to attach a large sugar molecule, a glycan, right on top of that decoy. This glycan acts like a physical shield. It's a kind of molecular camouflage that makes that distracting epitope totally invisible to your immune cells. And this all comes together in a systematic, three-step process called immunofocusing. First, scientists pick an epitope they want to test. Second, they introduce that glycosylation sequence to mask it with one of those glycan shields. And third, they measure how well the immune response is working using something called the neutralizing immunogenicity index, or NII. Now think of the NII as just a score for how effective the antibodies are. If you mask an epitope and the NII score goes up, great. You've hidden a decoy and focused the attack. But if the NII goes down, oops, you've accidentally hidden a critical target. Okay, and now we have to pivot. We've just seen how this toolkit can be used to protect us, but now we're going to explore the theoretical flip side, how these exact same principles could, conceptually, be inverted to engineer disease. The key mechanism we need to understand here is something called molecular mimicry. Really, it's just a tragic case of mistaken identity. It happens when a tiny piece of a pathogen looks almost identical to a protein in our own body. The immune system learns to attack the bad guy, the pathogen, but because of that similarity, it gets confused. It cross-reacts and starts attacking our own healthy tissue, which is what triggers an autoimmune disease. And look, this isn't just some random theory from a sci-fi movie. The idea of weaponizing autoimmunity actually has a historical precedent. We know that offensive biological research programs, especially in the former Soviet Union, looked into methods to induce chronic, self-perpetuating diseases. The whole goal was to create something way more insidious than a typical bug. And a key method they documented was inserting genes that code for human proteins directly into viruses, creating essentially a delivery vehicle for self-destruction. You know, our own bodies have inherent vulnerabilities that could potentially be exploited. Just take our nervous system. It relies on incredibly precise patterns of glycosylation to work properly. And we know this because even tiny natural mutations in the genes that control this process can lead to absolutely devastating congenital disorders. This natural fragility demonstrates a really terrifying principle. If you can figure out a way to deliberately mess with these critical glycan patterns, you've got a plausible and devastating path to inducing disease. And this is where you can really see the perfect inversion. The vaccine designer's goal is to ask which epitopes are distracting my immune system and then use genetic engineering to mask them. The theoretical weapon designer asks the opposite, which pathogen epitopes mimic critical human proteins and then uses the exact same tools to enhance their exposure. It's the same logic just applied to a completely opposite, destructive goal. So the whole toolkit gets turned upside down. Step 1. Instead of adding glycan shields, you remove the natural ones that might be hiding a self-mimicking sequence. Step 2, instead of adding complex glycans, maybe you engineer the pathogen to use simpler ones, making a dangerous area more visible. And finally, with tools we have now like CRISPR, You could precisely insert the genetic code for a human protein directly into a virus. All right, so let's move to our final section and see how this all builds to the central dilemma here. These two opposing goals, one for protection and one for harm, they both spring from the very same core capability. And this illustrates the whole issue perfectly. On the left, you've got prophylactic vaccine design, masking decoys, enhancing protection. On the right, you've got offensive pathogen design, exposing mimics, inducing autoimmunity. And right there, smack in the middle, they overlap. That's the shared foundation, the core capability of precise epitope engineering. The science, the tools, the knowledge, it's all exactly the same.
So the crucial point really is this paradox, and the source material itself says it perfectly. Rational design techniques, originally intended to make vaccines safer, paradoxically, provide the exact methodological and genetic tools necessary to achieve the opposite. At the end of the day, the tool itself doesn't care how it's used.
So this fundamental knowledge, this ability we have to understand epitope structure and perform precise genetic manipulation, can be pointed toward two profoundly different ends. The first is what we all want, the development of highly effective, safe vaccines. But the second is the theoretical construction of a highly specific stealth biological agent designed to cause chronic, debilitating autoimmune disease. And this leaves us with the critical takeaway from this entire explainer. We have developed an unbelievably powerful and neutral capability, but its application is anything but neutral. This highlights a permanent and incredibly challenging dual-use dilemma in biotechnology, and it leaves us with a question we all have to confront. The capability is here, so how do we as a society govern its application?